into the major classes of receptors, there are three: G protein coupled receptors, enzyme coupled receptors, and ion coupled receptors. Now, the G proteins are called so because they interact with G proteins. And the term G protein is referred to as G protein because they bind to guanine nucleotides, your GTP and GDP. G protein coupled receptors are receptors that span the cell membrane 7 times. Think of it in terms of a spring with 7 coils. The front end, that is the amino terminus, is outside the cell, and the back end, your carboxyl terminus, is inside the cell. The extracellular portion binds to the ligand, and the intracellular portion activates the G protein. Now, G proteins are made up of 3 parts: alpha, beta, and gamma subunits. The alpha subunit is where your GTP or GDP binds. So when your G protein coupled receptor binds to a ligand, it will cause an interaction with G protein in the intracellular unit. Moment this happens, your GDP is replaced by GTP on the alpha subunit. And this change will make the alpha subunit separate from the beta and gamma subunits. The free alpha subunit can now activate an enzyme known as adenyl cyclase. Adenyl cyclase makes a molecule called cyclic AMP, which acts as a second messenger. The G protein will stay active as long as the alpha subunit has the GTP attached. It turns off when the GTP is hydrolyzed to GDP, an inorganic phosphate. This conversion will change the alpha subunit's shape, making it reattach to the beta and gamma subunit, returning the G protein to its inactive state. Now, depending on their alpha subunit, the G proteins are further classified into 3 types: your GS or G stimulatory proteins, GQ, and GI. Now, your G stimulatory or the GS protein, when it is GTP bound, it activates adenyl cyclase, which increases the production of cyclic AMP. Now, cyclic AMP will move through the cell cytoplasm and bind to protein kinase A. Protein kinase A has 2 parts: a regulatory subunit and a catalytic subunit. So, the cyclic AMP will bind to the regulatory subunit, freeing the catalytic subunit, and the catalytic subunit can add phosphate group to the other proteins downstream, triggering a cellular response. Remember, all kinases add phosphate groups to subsequent proteins. Now, common GS receptors are beta adrenergic receptors, glucagon, histamine receptors, serotonin receptors. Now, in microbiology, the toxin from Vibrio cholera affects the cell by binding to the alpha subunit and stopping the GTPase activity in the intestinal lining. So what does this imply? It means your adenyl cyclase stays active, constantly producing cyclic AMP, and high levels of cyclic AMP cause the intestinal cells to secrete a lot of fluid into intestine, causing diarrhea. Now, coming on to GQ protein, which activates an enzyme called phospholipase C, which is present in the plasma membrane of the cell. Phospholipase C will cut the phospholipid called phosphatidyl inositol 4,5 biphosphate into 2 molecules, inositol triphosphate and diacyl glycerol. Both of them will act as second messengers in the cell. Inositol triphosphate is a water-soluble sugar phosphate that can move freely through the cytoplasm. It will bind to the receptors on the smooth endoplasmic reticulum and open the calcium channels. Now, the calcium concentration is higher inside the ER as compared to the cytoplasm. So the calcium will flow from ER into the cytoplasm. These calcium ions act as second messengers, binding to various molecules to trigger specific responses. Also, the increased calcium in the cytoplasm can change the cell's electrical charge and cause depolarization. Meanwhile, your diacylglycerol will stay attached to the cell membrane, and it acts as a recruiting center, if you remember the second pathway, and it will bind to an enzyme known as protein kinase C. Protein kinase C will then add phosphate groups to serine and threonine residues in various target proteins. Receptors for GQ proteins are muscarinic acetylcholine receptors, histamine 1 or H1 receptors, vasopressin, and serotonin receptors. Lastly, we have a GI or G inhibitory protein. Now, this will also bind to adenyl cyclase, but it will inhibit. So, essentially, your GI protein is opposing the action of your GS protein. You've met the G proteins, the cell's very own switchboard operators. They're quite busy swapping your GDP for GTP, like a fashionista changing outwards, and in turn activating enzymes like adenyl cyclase and phospholipase C, basically making sure everything is running smoothly in a cell. So leaving you with a mind map of G proteins for revision. Please like, subscribe and share. Thank you.